itself is not a receptor, right? That was the first thing. So they are receptors, and they're not named in the article because we're not exactly sure, but we know there's more than one receptor that can potentially work with the JAK pathway, right? So the JAKs actually sit, right? They're attached. Let's see them in a different color. They sit over here, and they're kinases. What does that mean?
They can phosphorylate each other.
That's right. JAKs can phosphorylate each other. Now, the common, this is where it's hard to draw in biology. These things aren't stationary, meaning a bunch of these JAKs just come and go, right? They're not like permanently attached. They just have an affinity for the receptors, which means they do, right, by like amino acid sequence, right? They do have a hydrogen bond type of affinity. So they basically kind of stay nearby, but they just kind of come and go, right? So normally these two pieces of the receptor are separate and they're far enough apart, and even though they accidentally come closer together, it's not long enough for the JAKs to come in contact with each other. Are you good with that? So these are just kind of floating. So when we get a ligand, right, which can be different things, but let's go with a cytokine, okay? And cytokine would mostly be in the immune system cells. Right? If it's not an immune system cell, it would be growth factor, okay, which we're going to go over there with rats. But a cytokine will come through, and it usually has, right, a site or a binding to both receptors, like this. And the binding of the ligand forces them, right, trying colors, right, it forces them to move closer together and stay there. Good with that, so that they're stable long enough. So as soon as that happens, right, we have JAKs that are now close enough to phosphorylate each other. So ATP just floats around everywhere, right? It's everywhere in the cell. It gets made all the time in the mitochondria, which we'll come back to in a little bit. So ATP, as soon as JAK is close enough, right, it phosphorylates, or dephosphorylates, I should say, ATP, making it into ADP, right, and adds the phosphate to another JAK. We're okay with that, and then vice versa. The other JAK does the same thing. So they cross-phosphorylate each other, and sometimes also the receptor itself, right? And that's what they said. Sometimes the receptor itself gets cross-phosphorylated. Not all. We're okay with that. So what happens then, right? STATs and PRB, right? That's from the other article, and ERK and MEK. These were all mentioned in different articles. Are all inactively floating around the cytoplasm, okay? So again, they all kind of bump into each other here and they're just diffusing. They're just floating, so there's no directional property to it. But once JAKs, because remember, they can detach or attach, are phosphorylated, bring this back over here, they can now bind, there's a binding site on each stat, which they cannot do if they're not phosphorylated. Okay, yeah, so phosphorylation always causes some kind of shift in shape, right? It's three-dimensional. So all of a sudden we have an axis or shape that we didn't have before, right? So as JAKs do then, right, we phosphorylate ATP, ADP, right? And we phosphorylate STAT, making it change its shape. Okay, hopefully this is all familiar. Into something else, right? Change the shape somewhere else. And then we need another stat, right? So another JAK will come in and do that. Keep it consistent. All right, here we go. ATP, ADP. There we go. We're good with that. So then this stat also changes its shape, and now they can physically fit together. We're okay with that. But before they just couldn't fit, it's like lock and a key, right? So now they become, let me see if I can, all right, with a P and a P, like that. Good? So in that shape is when they have the right code and shape to cross the nucleus. We agree with that. So if they're not dimerized, they cannot pass. So we have nuclear pores, right? And some of you mentioned the RAN pathway, right? Some of you mentioned that. The RAN pathway is a protein that basically drags things into the nucleus with the right code. That's what it is. I mean, it gets more detailed, but we don't need to know that. So here we go. So stats are now able to go, right, and basically open some genes. And one of the genes that transcribes are SOX. In the meantime, SOX are dormant, right? They're not made, right? And another thing it will do is also do some cytokines. It might do some growth factors, right? Anywhere it physically fits in the promoter, right? Promoter is the starting region. These are genes, right, that are on the exposed part of the DNA. Some part of the DNA are folded in, right, and then it has protein that this particular cell doesn't need. We're okay with that. So if this is an immune system cell, maybe this is a T cell or B cell or macrophage, right, and we now found an infection and they need to divide, right, because remember T cells and B cells make clones of themselves, we're going to make a bunch of cytokines and a bunch of growth factors to help it divide. Now this all happens with me crossing over to RAS, so stay with me for a minute, right? Do most cells spend any time dividing? No. So this is a little bit from the other part. So if you remember from 9th grade biology, right, we have the cycle, the mitosis part, right, which is a very small part of the division. But we have also G1 stage, S stage, M stage, and G2 stage. Is that familiar, right, a little bit? Yeah. Those are the stages of a cell growing and dividing. Do most cells spend any time in any of those stages? No. Most of your body cells are in G0, which means they don't go through any division at all. They're just normally living cells. Anyone know how many times your average cell divides in your lifetime? 40. Right. That's right. We did that before. Most, yeah, in your lifetime, your cells in your 90-year average will divide 40 times. And if we could live longer, let's say we figured out, right, we would all have cancer. Because the thing is, every time you divide, there's some mutations. And we'll talk about why it's seemingly there's more cancer. But cancer is inevitable, right? Sorry. We just die before usually most people get it. So there's a reason that we are getting it earlier and earlier, right? Because that's to do with DNA and mutations. But every time it divides. Now, obviously, your blood cells divide more often than that, and your neuron cells don't divide ever. Right? So there's sort of an extreme case. But most of your cells do not undergo division. So these immune system cells, your blood cells, get kicked into this much more often. So for example, they're going to have growth factors, and they're going to have all kinds of proteins that are CDKs, right? Your CDKs, right? That are open, where most cells would have those guys closed and not as exposed. Okay, so we're good with that. So here's the stat. Now, what happens then? So we're going to read, right? As this gets read, we make what? Right, mRNA. So we are reading these genes, right, and we're making these factors. mRNA, of course, is going to leave, right? Right, so it leaves the nucleus and gets read by the ribosome, right? The ribosomes are going to read the sequence of that gene. Okay, do colors here. And as they're reading it, right, they are assembling a chain, right? So they're going to fold it, right, and tertiary, secondary, etc. Eventually going to make a protein, right? So they're reading and putting amino acid sequence in the right order, right? Now, will these ribosomes go into the ER? Right, because we're reading SOX. No, we're not. Good. Why not? They're staying in the cytoplasm. So these will be read on the free ribosomes. Some of these ribosomes are probably making, some proteins like the GLUT1, the glucose, or receptors, or aquaporins, or whatever you just studied. Those are going to the membrane, right? But we're staying in the cytoplasm. So SOX, okay, and I put on some of your feedback, right? SOX are a negative feedback mechanism. What is that? And I wanted some of you to go a little bit more detail. Because some of you mentioned SOX as just, okay, they're negative regulators. Well, there are a bunch, there's several negative regulators, but they are actually directly as a negative feedback. Now, we did negative feedback back, I don't know which chapter, but what does it mean to be a negative feedback loop?
Reverse it, right?
Give me a little more. What reverses what? because to be it's just a specific thing. It's not just anybody reversing the pathway. Anyone?
It's like when we're talking about like it helps maintain the homeostasis. When you get too much of something, it tells you it like stops the production until you get back.
Correct. So, but what stops the production to make it an actual negative feedback loop? Because you're right. There's just regulators. There's regulators, but not all regulators are on a negative feedback loop. Anyone? Who regulates you if you are feedback loop? See, normally right. So let's think about the process. Normally, one guy tells another guy to make something. Receptors phosphorylate JAKs. JAKs phosphorylate JAKs. JAKs phosphorylate STAT. STAT, right, makes this. So it's a cascade, right? So we're making more and more and more stuff. So to be a negative feedback loop, it's the end product of that same pathway that shuts itself down. You with me? That's a negative feedback loop, meaning the pathway that you're actually making the product, we need more of. The product itself, when it gets too much, will shut itself down. Does that make sense? So when SOX gets made, and there's more and more SOX being made, SOX themselves are then going to come over here, right? Actually, more so, they're actually going over there. Right, they're actually going to go over here, and they're going to do a couple of things, right? They're either going to, they're attached to JAK, that's what they do, right? So SOX attached to JAK, and either physically sit there, like they physically block JAK from phosphorylating STAT, right? And the other part of what they do is by attaching, right? So let me just do whatever squiggle I did. Okay, SOX. By attaching to JAK, they cause a dephosphorylation, right? So they cause the phosphate to leave. So there's a bunch of phosphates then carrying it out. You okay with that? So that actually, so by JAK pathway, we make the SOX, and then SOX themselves, as they rise, they shut themselves down, right? They shut the production of themselves, and then anything else that was happening with stats. So cytokines stop, growth factors stop. So we stop, right? Because once a cell is ready for division, basically this is going to trigger it to go into a G1, right? So it's because it's in quiescence, it's in G0. So these guys are going to get made and throw the cell into a G1. Okay? Yes?
But how does the transcription, like the two stats that I'm thinking, know when to start making SOX?
So as soon as it's kind of like insulin bumping into it. As soon as they bump into a promoter that they fit, transcription starts. Right? Because remember, nothing is intentional. There's no communication. It's literally, I'm present, which means now a polymerase, right? So remember, polymerases, if you remember, are there to read that. So let's say we have this polymerase, right? DNA or RNA polymerases. So in this case, it would be RNA polymerase, right? Because we're making RNA. Right? So our RNA polymerases are floating around in the nucleus, but they cannot start transcription on their own. So they have either transcription factors that already sit there blocking things permanently, and we need somebody else to enter and take them off, and then all of a sudden polymerase can fit and read it. Do you know what I'm saying? Or this attachment, as it is not, polymerase is not able to bind to it, right? Because of its three-dimensional shape. So when STAT comes in, STAT has an affinity of whatever is in the promoter of SOX. Do you know what I mean? So wherever the gene starts. So whatever it is there, polymerase can't join. But when STAT comes in, STAT attaches to that part, and all of a sudden polymerase has a place to attach and start reading the mRNA. So as soon as STAT then disappears, right, and proteins, the transcription factors get degraded all the time. So as STAT stop entering, they'll get degraded in here, right? They have a short lifespan, and then they'll come back out and then die, right? So, and then polymerase will be stuck again and not able to read those genes, okay? So, go ahead.
So at this point, when SOX are already made and they attach the JAK, are like, what was needed to be made that they need like cytokines? Are those already made in the cell?
So again, so this is what's happening. So mRNA cytokines are also being made.
Okay.
Yeah. So anywhere that STAT can physically have, yep. So there'll be another one here, right? So there'll be another ribosome.
So it's
reading
not
this
just
mRNA.
SOX all the time.